Welcome to parasitology. This is the first lecture, the day one material first lecture. Starting with the definition of parasitology, this seems easy enough. Of course, it's the study of parasites, but what is a parasite? It is an organism that lives within, upon, or at the expense of another organism known as the host. So usually the host is suffering while the parasite gets to thrive. So they can live within us, or like, for example, head lice can be just on our skin. And the harm is not always extremely noticeable. It might even be subclinical, meaning the patient doesn't even know they have a parasite. But nonetheless, the parasite is using the host resources to thrive, and the host is losing something to the parasite. So this can be compared to two other terms which are also symbiotic relationships. So symbiosis is just a general umbrella term describing the relationship that two organisms would have. So parasitology is one of those descriptions. There's also a commensalism and mutualism relationships that exist as well. So in mutualism, both organisms benefit, and there's lots of examples of these in nature. I'll give you three examples. There's bees and pollen. So bees fly around, land on flowers, and they feed on the nectar and the pollen, and then the pollen sticks to them, and they're able to bring pollen to another area so that the flowers thrive there and pollinate other flowers. So the flowers get to continue their life cycle, and the bees benefit from the food. We have a mutualistic relationship with plants. So plants give off oxygen, which we need, and we give off CO2, which all the plants need. There's also our gut bacteria, which is part of our normal flora. So that bacteria gets to thrive, and we give it nutrients, and it sets up shop, sets up home in our bodies. And in turn, we benefit because the normal flora of the gut keep other bad bacteria from overgrowing. So when they inhabit our intestinal tract, we don't get overgrowth of like yeast or giardia usually. Other things that could harm us are kept in check. And that's true for most of all of the normal flora that exists in our bodies. And then there's commensalism. So this is when one organism benefits and the other organism is neither harmed nor benefited from the relationship. An example of this is birds hanging out on the backs of cows. So if you've ever passed by a pasture and seen birds just sitting, hanging out on the backs of cattle, they're waiting for the cows to feed on the grass and stirring up insects. And when they do, they can go grab the insects. And so it's easier for them to feed. Now, the cows are neither harmed nor benefited from this relationship. It doesn't matter to them that they're on their backs. They don't care, and they're not being harmed, and they're not gaining anything either. So that's just to orient you to what we're talking about here. Of course, our focus from here on out is going to be on the parasites. So you could spend your whole life studying all the parasites that exist. What we're concerned with are the medical parasitology topic. So this is the area of micro concerned with these invertebrate organisms capable of producing diseases in humans. So when the disease occurs because the damage is detrimental and noticeable and severe, when the host notices it, then it becomes, of course, a disease. So what's the big deal? Why do we need to study parasitology? Well, worldwide, billions of people harbor parasites, meaning They're carriers. They have them in them, with about a fourth of those people actually being diseased and suffering from the parasitic infection. This occurs mostly in tropical and subtropical areas, and we are in the southern United States in a subtropical area. It's nice, warm, humid climate. Parasites continue to be a leading cause of death worldwide. So, fortunately, we live in a developed country with access to healthcare and everything, and we generally don't see these outbreaks and deaths of parasites here because of that. But in other areas of the world, these are very serious problems. Still, 1 to 2 million people die per year of malaria. Leishmaniasis occurs mainly in Africa. That's a very detrimental disease there. Amoebiasis, caused by Entamoeba histolytica, is very severe. And the last major one is schistosomiasis, which is the schistosomes are the blood flukes that we'll talk about later. So while we may not hear about it here too much, and we don't, it is still a very serious problem in other areas of the world. So I have a picture here to remind you of where the tropics and the subtropics are, of course, on either side of the equator. So we're in the zone. We can definitely see parasites, and we do. We do see them. So why do we see them so much in our developed country? Well, infections are prevalent here due to the fact that we have immunosuppressed people who could develop an opportunistic infection. So immunosuppressed patients have a weakened immune system. These are like the AIDS patients, transplant patients, chemotherapy patients, and they are more susceptible to dying or getting severely infected from any infection with their weakened immune system. So for example, if you or I were to get a toxoplasma infection, toxoplasmosis. This might not be a big deal for us. We might not even know, but it could be very, very harmful to someone with a weakened immune system. Mosquitoes are a vector, and they can become resistant to insecticides, just like other insects can. And malaria can become drug resistant, just like some of our um, bacteria can become resistant to antimicrobials. Same thing here. With increased travel, you can travel to an area endemic for malaria and bring it back here. And then a mosquito here can bite you and transmit it to someone else. So that's how things get spread. With climate change, this could force the vectors to migrate to new areas. So also spreading it to new areas where it's usually not concentrated. And by vectors, I mean, we'll talk about several vectors later on, but examples are ticks, flies, and mosquitoes. So like I said, compared to other areas of the world, here in the US, we do have a lower incidence of parasitic infections due to several reasons. In general, we have access to good health care. Um, we have educated doctors, there's people know how to cook their food, we have good sanitation, we have toilets that flush and prevent it from going into the water supply, so we don't have to prepare our food from fecal contaminated water. We just have good education about, we know we need to wear shoes when we go outside and wash our hands before we eat. We generally have good nutrition. So we know not to eat something that fell off the ground, that gets cooked properly. In general, we have an absence of most vectors. So I mentioned the ticks and certain, like the sand fly, which transmits leishmaniasis, is concentrated to Africa. The tsetse fly and the tridomid bug for trypanosoma are concentrated in other areas. So because of the absence of those vectors, we generally don't see those parasites. So more of the same factors that affect geographical location of the parasites. I've mentioned a few of these things already. We live in most of the houses here are sound, structurally sound buildings for housing. We don't have straw houses or that just have clay for the ground. Those people are much more likely to get infected with parasites because of those conditions. We know to cook our pork to a certain degree. We know in general, fish or raw oysters or things can transmit parasites if they're not cooked. Some people in other parts of the world, it's part of their social custom to not wear shoes or to, cook not cook their food a certain way. So they could just be more prevalent, more susceptible based on those facts too. And in general, parasites don't typically live in dry weather or extreme temperatures. So places that are more humid, lower altitudes, these are where the parasites like to thrive. If you're way up high in the mountains where it's very dry, very high altitude, parasites generally can't survive there. Okay, as part of this overview lecture, I need to introduce this concept of the parasitic life cycle to you. So a life cycle, just like every living thing has a life cycle, these are the stages of development that a parasite goes through during its life. So this involves the growth and development, the reproduction, and how it's transmitted. And here's a very simple schematic of an example of a life cycle. So it might start out as an egg, then a larva would develop into an adult, adults would lay eggs, eggs would hatch out larvas, and so it would continue. So on to that, we need to talk about two important stages here. The first one is the infective stage of the parasite. This is the stage of the parasite at which it is capable of entering the host and continuing development within the host. This will be different for every single parasite. So for every single parasite we talk about, you have to identify the infective stage and the diagnostic stage, which is the next slide. So the infective stage might be, maybe a baby gets it in utero because it crosses the placenta. So it gets crossed from mom to baby. That would be congenital passage. Or perhaps you can only get infected with a particular parasite if you step on it and it penetrates your skin. Or maybe you have to ingest it in contaminated food or water. Or maybe it requires inhalation or a vector bite, a bite by a mosquito or a tick. Or maybe it's transmitted sexually or by a blood transfusion. So these are all examples of ways that you could get infected with different parasites, and it's unique for each parasite. Then there's the diagnostic stage. This is the stage at which we're going to identify it in the lab, the stage at which it may be detected in secretions, discharges, feces, blood, tissue, sputum, whatever, and identified by the pathologist or the MLT or MT. And like I said, this is going to be different for every single parasite. So when you identify a diagnostic stage, it's two parts. It's always the life cycle stage of the parasite plus the specimen type. So for example, eggs in feces, or larva in sputum, or trophozoite in urine. So to talk about this a little bit more, have an expanded life cycle here. And this is just an example. For each parasite, it's going to have its own unique life cycle. But just for the purposes of introduction and getting familiar with this concept, let's just talk through this example that I've drawn out. So let's say you have a male and female adult parasites living in your small intestine. So they would mate. The female would become fertilized and produce eggs. The eggs, she would release the eggs and they would be shed in feces. So I'm showing an example. For this example of a parasite, the diagnostic stage would be eggs and feces. That's when we would be able to diagnose or detect this parasite. So eggs get shed. For this example, I'm showing that the eggs are actually not infective right after passage. And this happens with some parasites, that the eggs have to mature in soil for about a month, where the larva will develop in the egg. So this would actually be the infective stage. It's when the egg stays in warm, moist soil for about 2 to 4 weeks. What this means is if you were to ingest eggs right after shedding, you would actually be breaking the life cycle because that's not the infective state. That egg would not be infective yet. So only after it develops, the larva gets a few weeks to develop, would it then be infective. So if a human comes along and ingests the developed egg with the mature larva inside of it, then they get infected. And you're asking how or why would you be ingesting soil or whatever? It's because you go outside, do some gardening, you don't wash your hands, your animal, your cat or dog comes in with, soil on its feet and you touch the feet and then you go eat something. So this is actually more common than you might think, than it sounds like. So we accidentally ingest the infective egg, and then inside of us, in the intestine, that mature larva would then hatch out of the shell, hatch out of the egg. So then we have this larva who would then mature into an adult, and we would get male and female forms. Female, they would mate, female lay eggs. So this is a good example, like I said, of a life cycle stage. Okay, so preemptively, I'm answering this question for you. Why do I have to memorize all these life cycles? Because I'm telling you, spoiler alert, for every single parasite, you're going to have to memorize the life cycle. It's important that we do this so we know how to break its life cycle. If we know how it infects us, well, then we prevent that from happening. We know where to cut the life cycle and prevent that from happening. It's also helpful so we can have educated, collaborative conversations with the physicians and nurses about the type of specimen we need. So if they're suspecting Giardia, we we know that, if they're trying to send us a blood sample for Giardia, well, we know that doesn't make any sense because it doesn't get into the bloodstream. We would need a stool sample. So that seems simple enough, but for some of them, they can get a little complicated. So we need to be able to have these types of conversations with the physicians so we can tell them what kind of sample we need if they've been misinformed or misguided. And so we know what we're looking for in each specimen. So speaking of specimens, we have a lot of different types of specimens we can receive for parasitology examination. So we're going to talk about all of them. The first and foremost, the most common, the one I have a picture of here, is a stool sample. So a fecal sample is definitely the most common sample we'd receive, although it's not the only one. We can also get blood. We can get whole blood samples to look for malaria or babesia especially. And then I have the third big category would be everything else, tissue and body fluids. So going through them, the first one would be urine. We could get a urine sample, and they'd be looking for probably one of two things: Schistosoma hematobium or Trichomonas vaginalis. We could get a CSF sample, in which case we'd be looking for Naegleria species or Acanthamoeba species. Those are two very serious parasitic infections. We could get a sputum sample, and we'd be looking for Paragonymous westermanni eggs or Strongyloides stercoralis larva or hookworm larva. We could get a vaginal swab. We'd be looking for Trichomonas vaginalis in the vaginal swab. We can also get biopsies like a liver or lung or muscle tissue. We could get a skin swab for Leishmania amastigotes, if it's a cutaneous or mucocutaneous manifestation. We could get a sigmoidoscopy specimen, a duodenal aspirate. We could even get a swab from an eye looking for acanthamoeba as well. So it's really varied, and if just like for micro, you can get virtually any type of specimen in the micro department. Same thing here. So I mentioned I might have said a few terms you weren't very familiar with, like maybe the sigmoidoscopy. So I have a picture here of your GI tract. So spend some time just, I know you've learned this before at some point, but refresh your memory about all these sort of more not commonly as mentioned parts, like the duodenum. So the duodenum is right here. It's the end of the stomach where it starts to connect to the small intestine. So we could actually get, through an NG tube, a duodenal aspirate. The sigmoid colon is the latter part of the large intestine before it gets to the rectum. So a physician might go and collect a specimen from specifically this area. So like I said, I'll refer to some of these as we go along, but just this is a good refresher. The last part of this lecture involves an overview of the classification of the parasites. So my biggest hint for you to be successful in parasitology is to stay organized. And this is most easily done by putting things into categories. So the first major category we have are the helminths. These are the multicellular organisms and the worm-like invertebrates. So when I say worms, we're dealing with the helminths. The helminths would be the scientific term, worms would be the more general layman's term for it. And this is, your helminths are on test 1. So all the overview and fecal analysis and the helminths all encompass test 1. Test 2 is the next major category, which are the protozoans. Within the helminths, we have 2 major categories. The first one are the roundworms. So that's the common name. The scientific name is the nemat helminths, or also called the nematodes. So the phylum is nemat helminths, the class is nematode. And those are also known as the roundworms. And I will try to say this over and over because you need to know all these terms. You need to know every word on this page. Roundworm, nemat helminth, nematode, and then the second category under helminths are the flatworms, also known as platyhelminths. So that's the other phylum. We have 2 phylums, nemat helminths and platyhelminths. And there's 2 classes that we'll talk about in the flatworms. We've got the trematodes, also known as the flukes, and the cestodes, also known as the tapeworms. Okay? Then the second major category, test 2 information, are the protozoans. So these are the eukaryotic unicellular organisms. And there's 4 major categories here: amoeba, flagellate, ciliate, and apicomplexa. And these are mostly categorized by their motility. So the amoeba have a pseudopod, pseudopodia, that's how they move. Flagellates, of course, have flagella that propel them. The ciliates have the hair-like projections, cilia, where they move sort of in a rotary motion. And then apicomplexa actually do not have a form of motility. So I've got your nice little schematic that you memorized back in grade school. So here's a reminder of it. Kingdom, phylum, class, order, family, genus, species. So keeping this in mind will help you study and stay organized, I promise. Every parasite is comprised, we name it by its genus and species. That's how we refer to it. So you will need to know, and you will need to know how to spell every single genus and species that's mentioned. For example, I've listed 4 different species in the genus Plasmodium. And if you didn't know already, Plasmodium causes malaria. So this is one of the protozoans that we'll talk about, and we will study all 4 of these species in the genus Plasmodium. The genus is always capitalized, the species is not capitalized, so you need to write that appropriately. And when you're typing it, the most correct way is they're always italicized. And if you're handwriting it, you underline organisms, genus, and species. So this is the end of the overview lecture. I hope it oriented you to the material and sort of just laid the foundation, laid the groundwork for everything to come.